Professor Chris Bystroff from Rensselaer Polytechnic Institute in upstate New York. He and I share a common interest with respect to concern about the growing population, climate change, the state of humanity with respect to sustainability. He, though, has a different academic gig than I do. He is a professor in the Department of Biological Sciences. We have sort of similar trajectories. We both came from California. We both got bachelor's degrees in chemistry at small liberal arts schools. And then we both went to University of California for our PhDs. And we both took jobs as faculty back in the Pleistocene, 1999. But he works in the Department of Biology. You were an X-ray crystallographer for a while. Now he's into bioinformatics, and he does vaccine design. And he uses PCR and CRISPRs and all that kind of stuff. So he's really a biologist, and he's going to tell us maybe a 1-hour story on a project on developing a contraceptive vaccine. I don't know what kinds of birth control people in this room are using or not using, but this would be an unprecedented kind of birth control that I think will be interesting for you to hear about, and it could be a very useful thing going forward. So without further ado, let's give Professor Bystroff a welcome.
Thank you. Thank you.
I'll let him take it away.
It's so nice to get an applause before I've done anything. So I don't have to introduce myself now. You know who I am, but you don't know why I am. So I'll start with that, I think. Contraceptive Vaccine Project is the official name on this project, which was funded by this organization here, whose name is blacked out because they don't want to catch a lot of flack for this. And it's the Grantham Foundation. Billionaire guru from Wall Street got a conscience, you know, and decided he wanted to spend his money to protect the environment. And he identified, I guess, this as the main problem with the environment, the number of people. So this is why, as you can tell, we are going to focus on decreasing that number. Just a little background on the number of people. This is human population on the planet going back 1,000 years. Doubling time in the year 1500 was about 700 years. If it stayed that way, we would only be up to maybe 1 billion by right now, but it didn't stay that way. And the doubling time in 1803, when we reached about 1 billion, was now 260 years. And if it stayed that way, then by now we'd only be at about 2.5 billion, but it didn't stay that way. In 1928, the doubling time had shortened to 125 years, and in 1987 it was down to its minimum, which was 37 years. It doubled from 2.5 billion to 5 billion in 37 years. That got to be scary, and in the 70s there was an outbreak of people talking about population. It slowly died down. The acceleration also died down too, which I would tell you in a different talk in the department colloquium. But for now, I'm just going to tell you why we want to do something about that and how we're going to deal with it. The why is statistics on women who were asked in the first 2 years of their child's birth whether their pregnancy was intended or not intended. Almost 50% of them say that that pregnancy was not intended. And population is growing at a rate of about 80 million people per year. And so about half of those numbers are attributed to accidental pregnancy. So something should be done about this accidental pregnancy. Moreover, the cycle of poverty is where having too many kids makes life more difficult for you financially. Having financial difficulty makes it more likely that you're going to have more kids. In this study that I have a table on the left where they did this survey of what they call the planning status of pregnancy. And they see a correlation between being wanted versus being unwanted. And an unwanted has two types, mistimed and unwanted. And family income, so you see a lot of unwanted pregnancy among poor people. Educational status, you see a lot of unwanted pregnancy among people that have not had a good education. And you see a correlation between unwanted pregnancy and children from unwanted pregnancy being of low birth weight, so they're not getting good nutrition. Could that be because they were unwanted? Could it be that maybe there's, on average, women are not taking good care of themselves because they didn't want the baby? So this is sad, and we should do something about it. So let me start with some of the thinking about this. You know the outcome. It's going to be a contraceptive vaccine. But what's on the table now for contraception? Here's just the icon view of it. Each of these represents a different way to prevent pregnancy. What's wrong with these right now? Why are they only giving us about 50% effectiveness overall? Well, let's go down the list. Some of these are last-minute choices. You see a cream, you see barriers. These are things that you have to do at the last minute. What are you thinking about at the very last minute? Maybe not going, and maybe things are happening at the last minute that are preventing actions that protect you. What else is going on? You have pills. Pills require due diligence, require every day you have to remember to take a pill. You have some methods that require surgery, tubal ligation, not very easily reversible, or going into the uterus and putting a T-shaped device. Also requires going there, and it's kind of invasive to do that kind of thing. And some things, did anybody guess what this little party popper is up here for? What kind of contraceptive method would be represented by a thing like that? I see some smiles, so I think you know. That doesn't work very well. Just simply pulling out before it pops is not reliable.
That's the coitus interruptus syndrome?
Not reliable, yeah, that kind of thing.
It's approved by the Catholic Church, though, isn't it? Oh, no, not even.
Yeah. I'll tell you the only one that really works, and it's represented by this padlock. Can you guess what that is?
Chastity.
Yeah, just don't do it. It works. It works in Japan. Timing it, that doesn't always work either. So, and this was turned into a joke by a sociologist named Martha Campbell. And she said, it shouldn't be like this, but the analogy of this is like having this unwanted delivery man bringing you a very expensive appliance that you didn't order, but is bringing it because you failed to not order it. You failed to call them up and say, don't deliver a refrigerator, please. We don't want any more refrigerators. And nonetheless, he brings you a refrigerator every year or two, even though you've got 3 refrigerators already, a boy and 2 girls. He brings you a refrigerator. You're expected to not only pay for it, but fill it full of food and send it to college after 18 years, right? Shouldn't be like that. It shouldn't be that you just have to keep stopping pregnancy from happening. So I'm not the first to think of this. I have to credit Eric Pianka for this idea of turning contraception upside down. He called this the Johnny anti-Appleseed solution. So he says, first, bless everyone with infertility, and he says, you could say design a molecule that binds DNA and makes people sterile, and then you can't just make that permanent. You got to make it reversible. So you design another molecule that unmasks the first one, and then you regain fertility, but just for a short time. I mean, how long does it take? You unmask it for 20 minutes. Okay, maybe a day. And then you, in Eric Pianka's conception of this, make people work for that second pill, or only give it to them if they're married. Then you'd get responsible parenthood, says Eric, the lizard man from University of Texas, Austin. No more unwanted children. You have children only if you take an active role in recovering your fertility. Puts control in the women's hands, by the way. Shouldn't be in men's hands.
Planned Parenthood.
There you go. Planned Parenthood, every child a wanted child. This is in Eric Pianka's essay, The Vanishing Book of Life. So I liked that. I used to, I still do, give talks on population, and I'll often talk about Eric Pianka's work. And when this kind of soaked in, I started looking for if anybody's doing research into what could be this pill that could turn off fertility and this second pill that could turn it back on. And I found this guy, Raj Naz, became a good friend. He's a obstetrician and a professor in the University of West Virginia Medical School, and he comes from India, where he worked with G.P. Talwar, a guy's now 95 years old, and has been doing research in immune-based contraception for many years now. And they actually are, in India, have a contraceptive vaccine that's in clinical trials. So I decided to talk to Raj Naz and say, I'm a protein guy, I'm a molecular modeler. Can I get into this field? This is late in my career, but I wanted to switch from doing this other stuff to doing vaccine design. And so I learned a bit about what is a contraceptive vaccine. And it turns out there's three types. Vaccines are all using the immune system, using antibodies or using T cells, but what you target matters. So there's the ones that target gametogenesis, and those are going to block the formation of the eggs or block the formation of the sperm. This is used in animals, and it's called immune spaying or immune castration. There's the form called that targets fertilization, and that's going to target antigens on the sperm. I'm going to keep using the word antigen, so if somebody doesn't know what that means, ask me now.
What's an antigen?
What's an antigen? It's the thing that I wasn't ready for this. It's the thing that the antibodies attack. It's the anti-something. It's a protein sometimes, almost always. Sometimes it's a carbohydrate. It's usually a protein, but it's your target. When you have the immune system, you have to present the immune system with the target. That's called the antigen.
And so with COVID, it would be the spike protein?
Yeah.
Yeah.
So the antigen could also be a fertilization antigen, which is our case, and sperm proteins or egg proteins, or proteins that are involved in the signaling process of fertilization. And there's a third way to do it, and it's called an immunoabortifactant, and it would be targeting the process of the implantation of the fertilized egg into the uterus, and that's the method that Raj Naz's associate in India was using, and that's the method that is now in clinical trials. They target this, it's called the Talwar Institute, if you wanted to look it up. Talwar, T-A-L-W-A-R, is the word for sword in Hindi, and he's the guy who's 95 years old and has developed an abortifactant vaccine. And it blocks implantation of the fertilized egg in the uterus. So I, taking advice, so he was my guide, and taking his advice, we decided to come up with a plan for a contraceptive vaccine that used that second method, anti-fertility, the fertilization process. So here's the summary slide of it all. We looked at sperm for good antigens, and we wanted the antigen to be on the outside of the sperm. In this panel B, you're seeing those two curvy lines. That's the membrane of the sperm. Upper part is on the outside, so that's exposed to the antibodies from the immune system. So we wanted something that was outside of the cell. We call that the epitope. We take that epitope and we want to put it onto a carrier, and this is panel D, that will raise high immunity, that will have a strong immune response. Viruses are great for that. A virus looks very scary to the immune system. So we plan to attach our loops to the surface of a virus, inject that into males or females. The sneak peek is that it doesn't work in males, it only works in females, so I showed you a female cartoon. And they raise immunity to it. That's expressed in the vaginal mucosa. The mucosa of your body express lots and lots of antibodies, especially in the vagina where you do not want to get infections, so you have lots of antibodies there. Now you've got anti-sperm antibodies down there. And so when the sperm invades, you have antibodies ready for it. And it must be reversible. So the way to reverse it is to simply swamp out the antibodies. So we'll make this epitope and we'll put it on a non-immunogenic carrier, something not like a virus, so it doesn't raise more antibodies, but it's there and it binds up the thing that would bind up the sperm, freeing up the sperm and allowing you to get pregnant, just like Eric Pianka's second pill, which would cancel out the first pill. So we were all excited about getting started on this. Took a few years because we couldn't find any money, and then I was at this population meeting and I was ranting about not being able to get money to do a contraceptive vaccine. And I think I did a really good rant because the guy behind me from the Grantham was the CEO of the Grantham Foundation, and he took me to lunch, and from there began the whole story. A little bit of Immunology 101. If I have a scary immunogen, what's it do? It excites the immune system. It excites it that you have the sleeping immune system, and an immunogen out there is we have innate ways of recognizing things that are dangerous to the body. A virus, the size of the virus and the fact that it has repeating units on it is recognized as something very dangerous. So it raises alarm in the immune cells. The immune cells then, through a process which I skipped over, leads to the secretion of these antibodies. If you want Immunology 102, I have some slides for that too, but it's not quite as simple as I make it out to be. Now you've got antibodies, and then when it sees that virus again, this takes on the order of 15 to 30 days to happen, but once these antibodies have been created, then they're there and they're ready. Actually, to make it a little bit towards Immunology 102, it's not the antibodies that are floating around, it's the cells that make the antibodies that are floating around. And then when the antigen shows up, those cells called B memory cells are just ready to spew out antibodies. And the antibodies attack, and the antibodies now coat the thing that it regarded as dangerous, and then the garbage collector cells, the macrophages, go around and eat those up. So we're going to take advantage of the immune system and raise antibodies against something of choice. So we're not giving it a virus. So as such, we're going to modify the virus. So we're going to have sperm proteins within the virus. My student says this is like a sheep in wolf's clothing. It looks alarming, but actually all it is little sperm proteins. So we put sperm proteins in a virus, a dangerous looking thing, do the same thing. What happens? It alarms the immune cells. The immune cells now spew out antibodies against the surface of this thing, which is now coated with sperm proteins. Then when a sperm comes along, those antibodies are there. They bind up the sperm and prevent it from swimming, in this case. That's the basics of the immune system. Then the next part of the story gets into the function of sperm and how this is actually working. It's not so simple to just take a sperm, raise antibodies against it, and pretend that you've got immune contraception. It doesn't work. Sperm are evolutionarily adapted against raising any immunity. If they did, we would not have survived. Imagine if every time sex happened, you raised immunity against it. Then you would not be born. We wouldn't exist. So we had to be very careful and find like a chink in the armor of the sperm. This is where I teamed up with Jinju Chung, who is an expert in sperm. And from her, I got this slide. We're going to target the tail of the sperm, and this is the structure of the sperm tail, sliced down the middle. You can see the flagellum in the middle, which is responsible for the swimming motion, and a bunch of other topics that I'm not going to talk about, which involves how the signal from the surface of the sperm tail gets transmitted to the motor proteins in the middle and creates the swimming motion. But Jinju suggested we target cat sperm. Her favorite protein and something that is probably constitutes a chink in the armor of this very well protected cell. And cat spur exists in rows along the main part of the sperm tail, and it's an ion channel. So it sits there, and it's somewhat unprotected because it has to be facing out, and it has to allow calcium to get in. And calcium getting in is a sign that the sperm is ready to act. It's called capacitation. This happens because if the sperm was swimming all the time, it would use up all its energy while it's still in the male. So it has to be kind of quiescent in the male, and it gets woken up when it's in the female, and that's called capacitation. So this molecule, cat spur, wakes up the sperm and starts its hyperactive motility and allows it to penetrate the egg. This is from Jean Jude's lab again, and this is looking in a microscope at the way that the sperm swims. And she, in her publications, has identified two ways that the sperm swims. There's one that is kind of initially, as soon as it's capacitated, and this is called activated sperm. They've now been woken up, they're activated. And the next one is when it encounters progesterone, which is a hormone, as it goes up the fallopian tubes. I've got to get my anatomy right. It's going up the cervix, up the fallopian tubes. It encounters a hormone that is secreted by the egg, and then it goes into hyperactivity. And this hyperactivity is triggered by the calcium. The regular activity is triggered by bicarbonate and changes in pH around the sperm, but the hyperactivity is triggered by calcium getting in through cat spur. And that hyperactivity is essential for the sperm to penetrate, I think on my next slide, the surface of the egg. The egg is surrounded by this chorionic coat, which is called the zona pellucida, or the hairy zone. And that zone prevents most of the sperm from getting in, slows them down, and only the first one that is hyperactivated can get through. And hyperactivity, you can't tell, but it's kind of a corkscrew motion instead of a sinusoidal motion, drills it through the zona pellucida, getting it to the surface of the egg. So Jean-Ju Chung showed that if you knocked out the genes for cat spur, you got sperm that just swam very normally, and they didn't get hyperactive, even when they were exposed to progesterone. They did not fertilize the egg. Those mice, these are called knockout mice, where you've taken away the gene, were totally infertile. You couldn't breed these mice. So, and you could knock out any one of 8 genes, it turned out. 4 genes that form the calcium channel, and 4 other mystery genes, which at the time she had no idea what they were doing. So she was documenting in the literature that there are these extra 4 genes, and we didn't know what they did. We found out a year ago what they do. But so at the time, now I'm going back about 4 years when we started the funding, and we didn't know what cat spur looked like. My job as a molecular modeler is to build model of what this molecule looks like. So this is my model for the calcium channel. And so based on this model, I predicted that the areas of the sequence that are shown in orange in the sequence alignment here, these are amino acid sequences, those are predicted to be on the outside. So they're facing where antibodies can go, and so they should be available to those antibodies. And down the middle is where the calcium would go. And if we figured if we could take this loop, put it on the surface of that virus protein, raise antibodies, and then the antibodies would be targeted to here, and the antibodies are big enough that they would block the channel. We were wrong, but that's what our idea was for how this would work. So we started this before the structure came out and told us we were wrong. We made quite a bit of progress. We had at least 4 different antigens that we made those, meaning that we ordered the gene, and when we had the gene, we grew it in bacteria, And the bacteria we're using just as the little factory to make protein. And we pop open the bacteria and you get the protein out. To test whether, and then we purify it. And these first two channels here are proof of purity. And this is electrophoresis. We separate all proteins in the bacteria after purifying them by charge. And it should have just one band, and these are pretty pure. And then we look at, this is called a Western blot, and that's to show that it's stained by a specific antibody that we purchased, we already had. And then we found out, it's kind of a side topic, but it's marking the protein that we purified, but it's also marking a whole bunch of degradation products, so we had a degradation product problem, but we fixed it. And then what we had to do was take that purified protein, and we had to find ways of having it assemble into virus-like particles. This was a lot harder than it might look. This shows success. This is electron microscope data, and it's showing this particles that are 60 nanometers in diameter, and they look like human papillomavirus, which is the virus that we're using. So, but we call them virus-like particles because they're not infective. They don't have any DNA in them. And so these are those scary particles that now have that sperm protein on their surface. The protein from the sperm is so tiny it doesn't change the size of these particles. So that was the hard part. So now, I forgot to say what was on the slide. We produce these things in bacteria. We call them HPV, human papillomavirus, virus-like particles. Produce them in bacteria because we're thinking ahead that this could be done at the industrial scale. So it has to be scale-upable because the market for people with a contraceptive vaccine is many millions. We have to be able to make a lot of this stuff. So we picked the cheapest, most easily scalable method. We chose something that's safe and is already FDA approved. How many people have had the Gardasil vaccine? Nobody? Or Cervarix? I'm surprised because a large fraction.
Cervarix, is that like a flu thing?
No, it's a anti-cervical cancer vaccine. You might have had it without knowing it.
Oh, my son had to do it. He doesn't know that he's had it.
Yeah.
The way to
So-
prevent cancer in women primarily.
In women, but it turns out men can also get something like it. I don't know.
But they can
So-
also give it to them.
Oh, HPV, yes. So anyhow, that means it's FDA approved, and so we figured it would be an easy path to make something derived from it and get FDA approval. And then finally, what we are going to claim now is that this is a reversible vaccine, following the idea of Eric Pianka of having a part B that can knock out the effect of the first part. So now I'm going to tell you how that second part works. So this is reversing the vaccine. So now what we're going to propose to do, and we're already doing, is to have a decoy. And the decoy is the same antigen that we're using to raise the antibodies. The same antigen that we're putting on those virus-like particles and raising the antibodies, we're going to put onto a non-immunogenic carrier. Our non-immunogenic carrier is called albumin. And albumin is really innocuous. It doesn't do anything. It doesn't raise any antibodies. So you put that on there, and then that is included in a vaginal suppository, let's say. So before intercourse, you'd get a vaginal suppository with what we're calling the enabler. And, and these would go in and bind up all the sperm. All the antibodies that would attack the sperm. So you've got this thing floating around, in a sense, protecting the sperm from the antibodies that you have from the vaccine. And so the sperm now is not blocked, the cat spur is functional, calcium gets in, it can get hyperactive motility, and you have approximately 24 hours worth of fertility from this insert, this enabler. Do we have any success? Well, we did. In the Jean Ju Chung lab, they do in vitro fertilization. And that's done by, on a Petri plate, you have one drop with sperm in it and one drop with eggs that have been harvested from mice. And you take one drop and you move it to the other, and you watch that for 24 hours. And if you see it progressing to the 2-cell stage, you know that that's a fertilization event, and they count the fertilization events. So in this first one is antibodies that are generic antibodies against other things, and using capacitated sperm, so sperm that have been exposed to an artificial vaginal mucosa solution are capable of fertilizing the egg, and they do, because nothing's stopping them. And then we tried with these antibodies that we had raised against a piece of the Caspar ion channel. That one of those 4 loops that I had mentioned in a previous slide. And so we mixed the antibodies with the sperm first, and then mixed mixed that droplet with the egg droplet, and then watched it for 24 hours. And if I blew this one up, you'd see they're all single cell stage. They have not progressed to the 2-cell stage, so none of them got fertilized. And then for good measure, Xiaofeng, the student that did this, tried it the other way and said, because I think somebody was asking her, well, what if you mixed the antibodies with the egg and then had the sperm come in? So she tried that. It didn't make sense because the antibodies are not against the egg. So basically this egg and the antibodies are kind of floating around, not recognizing each other. And then you add the sperm, and it turns out it doesn't work, and shouldn't. The sperm are kind of competing. Some of them are running into antibodies, and some of them are going ahead and finding the egg. And so you get kind of a halfway result there. But that's not the way it will happen. It takes a lot of swimming to get to the point where the egg is present. You have to get up to the fallopian tubes, and in that long trek, they're going to encounter lots and lots of antibodies. So we expect the real result is number 2 here. So in vitro fertilization, blocked by antibodies against Caspar. We went and did some mouse studies, and this is old now, but this was 4 different antigens that we tried. We had short names for it. We tried them in males and in females, and we even mated the immunized males with the immunized females, and found a couple of them actually worked. One we called C5. Later we found that P1 worked. This was what we did in this case was immunize mice after they're immunized in day 0, day 15, day 30, and then at day 60, the boys are put in with the girls, and then at day 75, we check the girls for pregnancy. And it's very unlikely to not be pregnant if you're a mouse and you've been co-housed with a boy mouse. So that was a promising result. And so it worked, despite the fact that we were wrong about the structure of Caspar. So in 2021, a lab in China, using a fantastic new method called cryo-electron microscopy, solved the structure of Caspar. Here's where the mystery of where those other 4 genes was solved. It turns out they're outside. They're the top, in this image, the 2 parallel lines here, that's the sperm membrane. This is the outside, and this is the inside. This are the 4 channel proteins that we thought constituted the channel. Turns out the other 4 proteins are up here, and they're dotted with these little black things. These are sugars, these are glycans. And the reason for these evolutionarily is as armor against the female immune system. The sperm is defending itself against the female immune system. With this pavilion, as they called it, of proteins, antibodies can't get close to the ion channel, and therefore can't. You could raise antibodies against sperm if you didn't have this armor on the sperm, called the glycocalyx, by the way. So this presented a problem. Why did one of them work anyway? Why did one of these channel antigens work anyway? And the answer is, when we looked at it, it was one of the ones that was not blocked by the pavilion presence. And then B, what do we do now? We've got something that only works halfway, it turns out. So we started targeting the proteins that are up here, and we found one additional antigen that worked. But we're still in the process. This is an unfinished story. We're still trying to find chinks in the armor, trying to find places on this huge molecular structure that are available to antibodies, and that whose antibodies will then block calcium from getting in. So, in short, we're going to the new structure-based strategy and the plan for a next generation vaccine design, and we're already on a third round of this, is to look at this catspersome, and the molecular level, we're taking an antibody, model of an antibody, and docking it like a ship, and you're seeing where this antibody can dock. And that's an indicator that that place where it can dock is a valid antigen. And I've colored some of these places here, and I've put these labels on them. C5, P8, C7, C8. These are our codes for valid antigens now on the known structure of the Catsper, now called the catspersome. And when we've identified what we think is a valid antigen, computationally we'll take that piece and we clone it into the protein that makes the virus. So this is the HPV-like particle. It has this 5-fold symmetry, and it has places on its surface that are friendly to the insertion of a loop. And so we'll put it into one of those, grafting-friendly positions. And then when we make that protein in bacteria, we pop open the bacteria, we purify that protein, we put it in the electron microscope to make sure that it's forming these virus-like particles, and then we inject the mice. The mice raise antibodies, and the antibodies go to that spot, just like we did computationally in the mouse itself. Those antibodies are going to go to that spot when they're co-housed with the boys, and sperm is introduced into the female mouse. The antibodies will be there, and they're going to block the calcium channel of the. I don't know if you can see it, but the protein has these holes in it. That's where calcium can get in and then go into the cell. So this is armor, but you know how in normal armor you have to have holes for the eyes, right? So there are places where you can get in through the armor, and that's what we've identified by doing this docking. We figured that calcium has to get in somehow. So let's find antigens, find some antibody handles around those calcium portals, such that now when an antibody is present, it sticks there and it blocks those portals and calcium can't get in. And that's the plan. So while this has been happening, fertility's going down anyway, interestingly. So I went and I presented all this data to the head of the Grantham Foundation, Jeremy Grantham, and had to really sell it, because he's going, isn't fertility going down anyway? Maybe I should save money and invest in something else instead. But I want to make a case for doing things that will help the population go down in a more humane way. Decreasing fertility is a humane way. War is not. Famine is not. So I hope I convinced Jeremy Grantham, and I hope he continues our funding, because I'm so much more hopeful now about the outcome of overpopulation than I was before, partly because of I'm participating in this process of getting fertility to go down. This is this is an infographic which I really love. the colors are time. And so going from red down to green is the more recent ones. And then on the x-axis is the fraction of the population. So it goes from 0 to 1. And then each of these segments, line segments, represents a country. And so countries with higher population have longer line segments. So it's this cumulative. So the total fertility is like the area under the curve. And so you can see that dramatically the area under the curve is going down. And what's responsible for it, if you drill down, is China, first of all. Here's the China back in 1950 to 55. Average family size was blocked by this part of the image, but I think it's like, it's over here. So this is the number of children per woman. And in China, in the 50s, you'd have, one woman would have on order of 6 children. In the 75 to 80, that was already down to 3. And in the most recent studies, which was 2005 to 2010, it's below 2, which is the replacement value. You have to have 2 children to replace the parents. You actually need about like 2.1 because there's a certain amount of attrition between birth and childbearing years. So, do we no longer really need a contraceptive vaccine? Because it's the problem solving itself anyway? And I argue that we do, because what we want population to go down in a humane way and not have to, look to a dystopian future of famine and war. So I'm very hopeful. I'm doing my best to participate in getting the population to go down. And I just had one more slide, which is the slide that my benefactor asked for, which is, when's it going to be done? It's also my answer to one of the most common questions I get when I give this talk is, when is it going to be ready? So I just made this slide, and it's my best guess. I think at this point in time, we are going to achieve close to 100% contraceptive efficacy in mice within a year, probably under a year. Our plan is by 2024, we're going to, actually maybe the end of next year, start work in macaque colony in Oregon. And so they're going to vaccinate macaques and then look for incidents of pregnancy there. That has to be done before clinical trials in humans can take place to establish safety and efficacy in a non-human primate first. And then around 2027, we expect we'll go to clinical trials 1. That means that volunteers in the clinical trials would take the contraceptive vaccine, but they would also have their own backup contraception. That would go to, if there are no safety issues with it, and I don't expect any, then it would go to clinical trials 2, where the number of volunteers goes up, and then they're testing the efficacy. So these would be women that would decide not to do any other contraception, just take the vaccine. If that is good enough, then it goes to market. The whole process could fail at any point. It could even fail after doing all of this because efficacy is only 80%. And who's going to buy a contraceptive that's only 80%? So we're hoping for the best. I have high hopes because my friends in mousy places believe that if you can block fertility in mice, you can block fertility in humans. But they're just way more fertile than humans are. So I have high hopes. So I would like to open the floor for questions on the contraceptive vaccine or anything else. Yes?
I do apologize, there's a good chance you said this already. How long did you say you've been working on this?
I got funding about 4 years ago.
Okay.
But we've been working on it for like 3 or 4 years before that, so it's about 7 or 8 years we've been working on it, but it was backburnered until we got funding can only do so much without any money.
But you still make a living as a faculty member, teaching classes, and you have
Yeah.
other projects going on. It's a long-term investment.
Yeah. Yep.
So, since it's a non-hormonal birth control method, will there still be side effects that like, or there likely to be side effects that are harmful for the person using it? Or-
Side effects are always a problem, and vaccines can have side effects, but this is modeled after an existing vaccine, which is already known to have no side effects. So, and the Gardasil vaccine has fought the PR battle against people that claim side effects against it. And they've proven that the side effects were chance occurrences. The so-called paralysis that came with the Gardasil vaccine is a myth. But so we think we're in good shape as far as side effects go. Yes.
So after you get the vaccine, would it last like until you get the reversal pill, or would you have to get it?
Yeah, that just pointed out a slide I forgot to include. Thank you. So I might have a long extended answer if you don't mind.
That's okay.
Okay, because this brings up a subject which I neglected. The market for a long-acting reversible contraceptive, so-called a LARC, is generally not permanent. They don't want long-acting to be permanent. And the market is, we're talking about the market being women of childbearing age, but you can divide it between young women who have not had families and older women or child-free women by choice. And the child-free or women that have completed their families want the permanent version of it. And women who, even though it can be reversed, the market is probably going to be such that they don't want a permanent reversible vaccine. So what we have proposed here turns out is going to be long-term, maybe even lifetime. Well, since that issue came up, we've been thinking of short-term immunity. And there's research on other immune carriers that provide short-term immunity. And so the slide I didn't include here has our cat spur being carried by a different carrier, and the carrier is called heat label enterotoxin, the short LTB for short. And the research shows that if you inject LTB with an antigen, you get the immune response to the antigen lasts on the order of months. It's almost too short. We're currently in the process of making our LTB version of the vaccine, and we've ordered the mice, and we're going to inject them in a few weeks with our short-acting, how do you call this? Short, long-acting reversible contraceptive. Short-acting? Maybe we should call it short-acting.
You need to work on that.
We have to work on the right acronym
Yeah.
for it, but we have evidence for it already that it lasts about 3 to 4 months, which is almost too short. So women, how long a long-acting reversible contraceptive do you want? I'll make it to order.
Yeah.
IUDs are currently years for contraceptive.
IUDs last forever, don't they?
Until you take it out, right?
Until you take it out.
Depending on the IUD, so like some are hormonal, like those ones don't
Yeah.
last forever, so like certain parts of the home, because those are also used for medical purposes, so
Right.
like if you're trying to have like less period, like period pain and stuff like that, that lasts about 5 years. With IUDs as a contraceptive method, I believe last 8 years, depending on the kind of IUD. It's more than one.
I would have to work with the immune system, and the immune system is not too flexible. It seems like the immune system's okay with immunity that lasts 3 months, maybe 6 months. And the immune system is okay with immunity that lasts for life. But there's not much room for fine-tuning in between. I don't think we have much chance of making a 5-year contraception that suddenly clocks out after 5 years, unfortunately. But we might be able to make a vaccine that you can boost. And you can get 3 months, and then get a booster, get another 3 months, get a booster, get another 3 months. So,
Is this injected? in
yeah,
blood? Okay.
intramuscular. Yes.
So, my understanding of antigens, you can have them that look like Phillips, some that look like a standard, some that look like a hex of nut, so it's like different shapes.
Yeah.
And you build things that get spit out by a cell that are antibodies, and they kind of just gum it up. And they just glom onto that shape, and it's a geometric thing. And once it's gummed up, it ceases to function, and the macrophage just comes and eats it, digests it, and spits it out as nutrients again.
Yes. Yes.
Is that a simplification that is okay?
What you're talking about is when it's against a disease in the blood. Yeah, that's what happens. It gums it up. It's called opsonization. It's labeling it for digestion by the macrophages. Yeah, but so antibodies don't always turn off the function of the thing it binds to. It always gums it up. Doesn't always turn it off. And a sperm cell with a gummed up cat spur still works. So it's not enough, unfortunately. We thought we'll just target antibodies to any position and it should gum up the works. It doesn't. They hang on to the antibodies and they just swim along just fine.
But they don't do the spiral,
Some of them do. And
So you have, the macrophage is actually what makes it ultimately work?
No, well, they're probably too big to be engulfed by a macrophage. They're a whole cell.
Right.
And also I think that's too slow. What's the analogy here? So let's say you've got a whole panel of buttons. Each one is an antigen. And you got one red one. And you have to find, and the red one is the antigen which if you hit with an antibody, it shuts it off. And the other ones are decoys. That's what the sperm is like in analogy. I just made that up. I think it's a proper analogy. There's a whole bunch of places where antibodies can go, but most of them, antibodies are just going to go for the ride. Does that answer your question? There's more to the story if you want. So we're going to have two flavors of the vaccine, the long-acting and the short-acting. Both of them are reversible. Any other questions?
Well, thank you, Professor.
Thank you.